Let's start, so please be quiet. And today is another more introductory class. We're going to talk about DNA and proteins and basic stuff. So I guess for some of you it will be a little bit too basic, but for others maybe it will catch some gaps that you have. Please make sure that you ask if anything is unclear, because the purpose of this class is to have everyone understand the basics of biology and neurobiology, okay? And don't be shy, okay? So we'll start. We'll start with some words about nucleic acids. So nucleic acids are the building blocks, the basic building blocks of life, of DNA and RNA, and they are comprised of nucleotides. Sorry, okay. Nucleotides are organic compounds. Remember, we were talking a little bit about that. Remember the periodic table? I hope you remember. Okay, so it's an organic compound that has a nitrogen base, a sugar, and a phosphate. So here, this represents the base. This is the sugar, which can be a ribose or a deoxyribose. We'll talk about it. And together, they make up a compound called nucleoside. Now, together with the phosphate, it forms a nucleotide. So these nucleotides are like letters in our DNA. Each of them is a little bit different, and we'll talk about that. And generally, these are polymers. So it's a large molecule, also called macromolecule. And it's a repeating structural unit. OK. So this is the same thing. Here you see the nitrogen base. You don't have to know the structures. This is the sugar, and this is the phosphate group. And this is the nucleotide. And we have different kinds of bases that give each nucleotide their identity. So we have cytosine, C, thymine, T, uracil, which is only in RNA, and we have adenine and guanine, A and G. So maybe you heard of these letters in the past, and if not, you will know now. So what is DNA? You probably heard a lot about DNA. What is this thing? Anybody?
It's a large chain of nucleotides which contains our genetic information,
right.
All our characters.
Right. And OK, great. So each cell has DNA in it, and it's a chain of nucleic acids. And it has the genetic information, which is packed in a very certain way. So the DNA is a double helix, which you probably know. It is located inside the nucleus. So this is, just to remind you, the nucleus is in the most likely in the middle of the cell, depends which cell. It has a membrane. It has pores. Why does it have pores?
So things come in and out.
Like what?
Nutrients come in and...
So not exactly nutrients. Nutrients are coming in and out of the cell, and they're usually going to the energy producing place in the cell, which is mitochondria. OK, what is that information?
the RNA.
The RNA. OK. So RNA is getting out of the nucleus through the nuclear pores, which are right here. And they get outside of the cell. And inside of the nucleus, we have the DNA. And RNA is traveling in and out, mostly out. And why is it going out?
To make proteins.
Right. OK. So what is the difference between RNA and DNA? So it has chemical differences, and it has functional differences. So chemically, we have some different characters. For example, the RNA has a sugar that is a little bit different than the sugar in the DNA. So we have ribose in the RNA. So this is ribonucleotide acid, nucleic acid. And here we have a different sugar, which is right here. Okay. Another difference is that RNA is single-stranded and DNA is double-stranded. What does it mean?
It means that the RNA can bind with another.
Right.
The DNA helix is completely closed off to anything else.
Exactly. Okay. So DNA is a very packed molecule. It is double helix. It speaks. Each helix is speaking within itself. And the RNA is more open and more available. And why is it more available? To allow transcription, to allow proteins to form. Okay, the next thing is the bases. So we have A, adenine, T, C, and G, which are all nucleotides. But in case of the RNA, we have U instead of T. It's a little bit different chemically, but this is not important for this class. And, but know that if you see a molecule that contains U in it, it's an RNA molecule. And if you have T, this is a DNA molecule. Yeah, this is the sugar. So here you see the ribose, and this is the deoxyribose. So deoxy-DNA, this is the D where the D comes from. And the RNA, this is where the R comes from. And this is how it looks like. So this is a very classical structure, double helix of the DNA. And here you see how the bases are pairing to each other. So we have A and T that pair with one another. And these bases are called complementary. So A binds with T, and C binds with G, as you see here. Is this clear? Okay. So A cannot bind to C or to G. This is the base pairing, and it is always like that. Now, between A and T, we have two hydrogen bonds, two bonds, as you can see here. And in case of the C and G, we have three. These are the hydrogen bonds. So would you expect some differences if a DNA contains a lot of C and Gs or a lot of A and T? So if we have different number of bonds between them, this will suggest how strong is the bond between the strands of the DNA. So which one will be more strong?
One that has more C's and G's.
Right. Okay, so DNA that is rich in C and G, which is called, we call it GC content, will form much stronger bonds between the strands. And it will be much more difficult to dissociate them, to dissociate these bonds. Okay. Another thing is the direction of the DNA, of the strands. So we have two ends. We have 5' end and the 3' end. And how does it work? So we number each atom in the sugar molecule. So here, this is number 5, and it goes 5. Here we have another one, 4. This is 3, 2, and 1. And this is the direction of the strand. So it goes from 5' to 3'. The last one here is 3'. Okay?
What does that mean?
I'll get there. And here it's the opposite direction. So the parallel strand goes from the other direction, from 5' to 3'. This is 3. And what's important for you to know is that it always binds in anti-parallel manner. So we have 5' that is talking to the 3' end, and vice versa in the other direction. Okay?
Anti-what?
Anti-parallel. Okay, so when we are talking about a sequence, we write the sequence, we always write it from 5' to 3'. This is the direction of the sequence of the DNA. So if someone is, if we're sequencing in the lab some DNA fragment, or we're interested to know more about a mutation in someone's genome, we can sequence it and we write the sequence from 5' to 3'. If we write it in the opposite direction, we get a totally different information. Okay? Yeah. So here you have a little bit of an explanation about the direction. So a phosphate group is bound to the 5', and the sugar binds to the phosphate in the 3'. And it goes all the way along the strand. And as I already said, so T and A's, thymine and adenine, have 2 hydrogen bonds, while C and G have 3 hydrogen bonds. OK. Now, this is really important. So RNA can interact with DNA. But DNA will not interact with a different DNA molecule. Okay? And this is due to the fact that this is a single strand, unlike the DNA, which is double strand. So just to summarize this part, so our genetic material has 4 important functions. It stores our genetic information in the DNA molecule. When it is expressed, the DNA can be expressed, right? And this is why you have dark hair, dark eyes, or whatever features you have. This is called phenotype. When DNA expresses and gives you some identity, this is the phenotype. The DNA can be prone to mutations. It is susceptible to mutations. So UV light, for example, or other different chemicals and other things can cause mutations in our DNA. And this can be really, really serious. We'll talk about it. And when a DNA, when a cell has to be replicated, so cells are getting old, they have to be divided and form new cells, the DNA duplicates. This is called replication. Okay? Any questions so far? Okay. So the DNA structure was first discovered by James Watson and Francis Crick. They got a Nobel Prize for that, and it was in 1953. There was a woman who participated in the study, Rosalind Franklin. She never got a Nobel Prize. She was actually Jewish, and it's an interesting story that they collaborated with her. They worked with her all along the way, but eventually she was not recognized for that. And now we go back to the genotype and phenotype. So the DNA, the genome, is the genotype. Each of us has different, we look a little bit different, we are different, okay? And where does this come from? So we have different DNA sequence. Now, most of the DNA sequence is very, very similar. So about 99% point something of our DNA is identical. And this is why we're humans. But we're still very different. And this comes from different genotypes. So there are like little flags along the genome. They're called SNPs, single nucleotide polymorphisms. And these are the nucleotides that give us our identity. So they can define your hair color, your eye color, or any other thing you can think of. OK. Now the phenotype is what comes out of the DNA. So in this case, this is an example of an Alzheimer's disease brain. So what do we see here? Do you remember last class? Yes. So we were talking about Alzheimer's disease. And what is this? It's a plaque. What is the plaque? Remember? So amyloid plaque. Sounds familiar? Amyloid beta? OK. So amyloid plaques are proteins that are deposited in the brain due to many reasons. Sometimes we think it's beneficial, but sometimes something it's protecting, something that this is the disease, this is the reason for the disease. And these are proteins that are misfolded. Okay, rings a bell? What are these? These are the tangles. What are these tangles? So these are proteins inside the cell. Okay, what are they? What do they do? Yeah, that's what happens in the disease, but in the native form, what they do is to give a structure to the cell. They are microtubule-associated proteins.
To unfold, though?
So in the case of the tangles, they're not unfolding. They're hyperphosphorylated. They have too many phosphates in them. Okay, but in case of the plaques, the amyloid beta plaques, they are misfolded. So this is the phenotype. Okay, and a little bit about the DNA replication. So another Nobel Prize, Arthur Kornberg, he received the Nobel Prize for unraveling the mechanism of DNA replication. And actually, his son got another Nobel Prize for the following process. I will get there. So this is Roger Kornberg. And so what is the replication? We have this DNA. So this is not a double helix, but in this case, it's open. Okay, but if we'll have a lot of it, it will form this helix. And when we have to duplicate the DNA, because the cell has to divide, and we have to pass the information to the new cell, this is how it happens. So the two strands, the two double helix, they open up. Okay, so this is called the template strand, the red one in this case. And on each and every one of these, we have a new strand that is growing. And this is how the replication works. So the hydrogen bonds, remember them? The bonds between C and Gs and A and Ts, they're opening, they're broken. And this way, each strand can form a new strand on the other side. And afterwards, when they're formed, they form the new hydrogen bonds, and they get the double helix structure. Any questions? Okay. Yes?
What breaks that bond? Did you discuss that?
I haven't, and it's a good question. So we have different enzymes that do that. But other things can cause that too. For example, in the lab, when we're trying to do that artificially, we just heat the DNA. So when DNA is exposed to heat, the hydrogen bonds are opening, are broken, and then we can access the DNA more easily.
What temperature?
Oh, it depends. So if we have a lot of C and Gs, it will be higher temperature than if we have a lot of A and Ts, because of the number of the hydrogen bonds.
The connection is more
strong.
between the cells.
Right. Okay. So here you see how the growing strand, which direction does it take. So this is the template strand. In this case, it goes from 5' to 3'. And we have the growing strands that grows anti-parallelly. So we have it starting from the 5' end, which will base pair with the 3' of the template. Okay? And it will grow in the 3' direction. So each time we add a new nucleotide in the 3' end.
What is the 3' and 5'?
So the 3' and the 5' are numbers of carbon atoms on the sugar. Okay? And it's like we need a map. As biologists, we need a map. We need to know where are we in the DNA. So we number the atoms. This way, we know where are we going. In the chain. In the chain, in the DNA chain. Okay. Yeah. So each time the chain is growing, we have a new nucleotide that enters here in the 3' end. So in this case, this is the new one. It will bind to the previous, to the last 3' nucleotide, and then it will base pair with the template strand. And as you asked there, so we have an enzyme that does that. It is called DNA polymerase. And we have different DNA polymerases. In this case, number 3 adds the next nucleotide here and binds the chain right here. Okay, so this phosphate will bind to this sugar. And this way, the chain will continue growing. Yes?
You said it's a double strand, and when it breaks, it starts building from 5 to 3.
Right.
So if on the side of the 3, it's okay, but if on the side of the 5...
It will never grow there.
Yeah, but you said it when the cell divides, it
right.
multiplies the DNA, so the 5 end of it gets...
Right, but so it's like each strand goes to the opposite direction. So this one goes this way, and that one is that way. So they break, and each one will have a chain growing in the opposite direction. Okay. So the phosphate bonds are broken in this case. Okay. And so we have this, each nucleotide comes with 3 phosphates. This is what gives the energy for the bond to form. So bonds are not just forming, covalent bonds in this case, are not forming without energy investment. So the cell has to put energy to form this bond. Okay? So it gets that from the 2 phosphates, and this is why it's important to eat phosphate. Okay? For example, fish has a lot of phosphate in them. And then these 2 phosphates are gone, and this phosphate is binding to this sugar. Yes, you wanted to ask something.
All of this DNA has like lots of genetic information, but what is considered like the unit of information? Like a sequence of these bonds?
Yeah, that's the sequence. So these are the letters.
But like the A and T bond is formed millions of times over. So
right.
You can't just say that is like one genetic information.
No, no, no. The genetic information is the sequence of each strand, not the bonds between the strand, but the sequence. So in this case, it's T, C, A, C, C, which will form here.
So there are like, you're saying 5 bonds that make up one, or is it just like the...
What do you mean by 5 bonds?
I mean, I'm trying to understand like where they actually recognize what the genetic information is. Like how do they say like, oh, this like humans have 43 genes, but what is...
Okay, so this is why we have the sequence. So each gene is made of these strands, okay, of a huge, not like this. Here we have only like 5 nucleotides. So a typical gene is a few thousands. So if you have a few thousand letters that are these nucleotides, so T, C, A, C, this is the sequence.
And all of them together make up.
Right, but you have a very, very long chain for a gene.
So it's
okay?
Not just 5 and 6.
No, 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 no. It's thousands, and sometimes even more. Okay? More questions? Yes.
Yes. What's the purpose of the replication process of the DNA? Why does it need to break up and then add new strands to match the 2 broken up strands of DNA?
So when the cell is dividing, everything has to duplicate, including the DNA. This is where your information is. A cell without a DNA just cannot form. It cannot work. It has to have a DNA. There is only one case of a cell that doesn't have a nucleus with the DNA. Do you know what cell is that? No? No, that would be really bad. So these are blood cells. Blood cells don't have a nucleus. They do initially, but then they lose it. Okay, but this is the only case of a cell that doesn't really have DNA in it, at a certain point, of course. Okay? Sperm, if a sperm wouldn't have DNA, then it would not be able to form an embryo later on, and that would be really bad. Okay? So, more questions? Okay, is this clear?
Sorry, you said that blood doesn't have
blood cells.
blood cells, but they are able to tend
Yeah, but these are not, okay, so our blood contains many different cells. Okay, and the red blood cells, which are called erythrocytes, lose their nucleus. Okay, but we have many different other cells there. Okay, so this is the central dogma of molecular biology, which honestly is not really updated nowadays. When I used to be an undergrad student, it was, but now it's not anymore. So what we used to think is that the DNA molecule is, which can replicate, as you see here, this is DNA synthesis, also called replication in case of dividing cells. But when it's transcribed, it forms an RNA molecule. So this is true. Now, RNA molecules can be synthesized, can be translated to a protein, but this not always happens. So in case of microRNAs, which Mona was telling you about last class, these are RNA molecules that are not coding. They are not forming a protein. Okay, they are left as RNA molecules, and they regulate different other RNA molecules. And you also have a paper in Hebrew that I uploaded on your course website that explains this more broadly, okay, about microRNAs. And if anyone is interested in an English paper, just let me know. Okay. So RNA synthesis, transcription. So each RNA molecule is the messenger. This is what, let's say, passes the information from the DNA and allows the cell to form a protein, which is the functional unit. Okay, so our proteins are those who execute whatever the cell has to do. Okay, and in this case, you see these are DNAs. Okay, it's the double helix. And in this case, it opens up, as you see here. This is called a replication fork, but it's not so important for you. And we have RNA, that the yellow molecule, that is forming, is building on top of the DNA. Okay? And it is further processed. This is called pre-mRNA, pre-messenger RNA. And it is then processed. We add some molecule on 5' end. On the 3' end, we have a poly-A tail on the 5', which is a lot of A molecules, a lot of A bases. Okay? And this is what allows the signal for the cell to know that, okay, this is where it ends. When we have a lot of A's in the end of the DNA, this is how the enzyme knows, okay, this is the end of the gene, I can leave. This is a signal. So these bases are information molecules, and they're also signals. Is this clear? Okay. And when this RNA is getting to the cytoplasm outside of the nucleus, it happens through, what are these? These dots here? Pores. These are the nuclear pores. So this yellow molecule, the mRNA, gets out of the nucleus through these pores to the cytoplasm. And here it is translated to a protein. And this happens by the ribosomes. These are the blue round shapes here. And the growing blue strand is the protein. Now, there are a lot of Nobel Prizes in this scheme. This is one of them. This is Roger Kornberg that was awarded the Nobel Prize for a translation. And by the way, the ribosome, I think I have it later on, but the ribosome, you know who understood how the ribosome worked? Have you heard of Ada Yonat? Yeah. So Ada Yonat is a leading professor in Weizmann Institute, and she is the one who deciphered how ribosomes work. So these are these guys here. We'll get there later. So this is the DNA molecule again. Each color represents a different base. So in this case, the greens are A, and the red ones are C, for example. Actually, you have to count the bonds. So here we have 2 bonds. So which bases will these be? T and A, and the green and the red,
C and G.
C and G. Not everyone together. Okay, and when the DNA is transcribed, we have the RNA molecule, which are single-stranded, you see right here. And okay, when it is transcribed, we don't only have one copy of RNA from each gene, we have many. Okay, so each DNA, each gene, will give rise to many RNA molecules, like here. Okay? So a little bit about protein synthesis. So this is the ribosomes that are responsible for that. And this is the translation process. So each mRNA, right here, this is the RNA. It goes out through the nuclear pores. This is the cytoplasm. You see it's very long in this case, but this is much, much shorter than the reality. Okay? In real life, this is a really, really long chain, thousands of base pairs in the most optimistic case. Okay? It can be tens of thousands and even more. It's a very, very long chain. So we have a code, a genetic code. And each 3 bases, A, T, and G, for example, they're a code for a specific amino acid. And each amino acid, so amino acids are the building blocks of the proteins. Many, many amino acids form a protein. And this is how it works. So these are sequences, and why do we have U's here? Because it's RNA. Okay, so the RNA is the messenger of the information. This is why it's called messenger RNA. And we have different sequences that are codes for different amino acids, the building blocks of proteins. So this is the matrix of the genetic code. And as you see, we have different amino acids. So each of the colored circles represent a different amino acids. And we also have stop codons. So why do we need these?
When we get to an end of a protein chain.
Right. So how will we know when the protein is ended? Where is the end of the protein? We need a code for that too. And the code is UAA, UAG, or UGA. These are the 3 options. Each of these will cause the building of the protein to stop. And that will form a single molecule, a single protein molecule. Any questions about this part? Yes?
What is the AUG? It's highlighted.
AUG, AUG.
It's met.
Yeah. Where is that? Where is that?
First
Yeah,
column,
here.
second.
Right. So this is a start site. So proteins always start, always, by AUG. This is a single amino acid that always starts the protein. As we have a stop codon, we have a start site. But it's not so important for you. And this is how the amino acid look like. So we have different amino acids. We have 20 of them. But we have a little bit more because some can be modified in different ways. But generally, we have 20. And they have different characters. So some are more soluble. They like water. They can mix there very well. And some are not. And why do we need that? So this amino acids, for example, with this structure right here, they don't really like water. They're hydrophobic. They don't like water. Why do we need that? So try to think, where do we have the proteins? Where do they found in the cell? Okay, so some proteins are in the cytoplasm, but other proteins can be inside the membrane. And what is the membrane? It's the barrier between the cell and the outside. Remember? And how does it form a barrier? It has lipids, it has fat in it. So in order to have a protein inside the membrane, for example, a channel that has to allow, for example, sugar in the cell. It has to be stuck in the membrane. How will it stay there if it's basically lipid, if it's oil, it's fat? We have to have an amino acid that can live inside this area, in the fat. And this is why we have hydrophobic amino acids, okay? Because they have to function in different places in the cell. And this will be more enriched in areas of membranes, for example. Okay? Or the other option is we have to fold the protein, right? And how will it fold? It can be folded in many different ways. But usually, the hydrophobic parts of the protein, which contain these amino acids, are stuck inside. So they're not exposed to water, and they hold the structure of the protein. Okay? Is that clear? Because you seem a little bit confused. So ask. Just ask. I'll repeat that. So we need hydrophobic. Okay, so these are amino acids that dislike water. Okay? They like to be in a different substance that is more oily, in lipids. And where do we have these lipids? We have them in the membrane, in the outer layer of the cell. We also have it inside, but it doesn't matter now. Okay, and sometimes we need proteins that are stuck in the membrane. For example, channels. So they can be glucose channels for sugar, or they can be ion channels for different ions to allow electricity or chemicals or this kind of things. But they're like a gate to the cell. Okay, so this is the gate. The gate is formed in the membrane, and it allows things in and out in different ways. Okay? And proteins that are found in the membrane are rich in these amino acids. Okay, you don't have to remember the names, just remember the concept. Okay? Is that clear now? Okay. So this is an example for an hydrophobic amino acid, and these are hydrophilic amino acid. You don't have to remember which is which. It's not important for your class. But hydro, water, phobic, hating, and philic, loving. Okay, hydrophobic, hydrophilic. Okay, I guess some of you know a little bit of Latin, so maybe it's familiar. And we have the tRNAs. So what are the tRNAs? These are RNA molecules that carry here the amino acid. So these are the workers that bring each amino acid to where it's supposed to be on the mRNA. So here we have a codon, for example, CCG. Okay, or in this case, it's upside down, so it's GCC, because this is the 5' and this is the 3'. So it has to be read in this direction. Okay? So we have to find the amino acid, the right amino acid to bind there and to form the protein. And what does that is the tRNA. So here it has base pairs that can bind to the mRNA. And then we have the amino acid that is bound to this tRNA molecule, and it forms the chain that you've seen before. And this is how it looks. So this is the mRNA molecule, and each 3 bases are a codon. Remember, we have 3 bases. And for example, these 3 bases are a codon for this amino acid. So this is the tRNA. It gets in the ribosome. The green part is the ribosome. It gets into the first position of the ribosome. It brings with it this number 3, amino acid number 3. And then it binds the previous amino acid that has been there before. And then we have the next codon, the next 3 bases. And we have another tRNA molecule, which is in light blue. And we have the corresponding amino acid. And this binds to the, okay, so it goes here. This binds to the growing protein, the growing chain of the protein. And then this tRNA is released and kicked out, as you see here. And it moves. Okay, so each time this ribosome, the green part, moves 3 bases. So it finishes working here, and then this whole part moves, and every time, every 3 bases, it keeps moving on the RNA. This is how it grows. It's like a machine. Okay? And yeah, so in this case, we have the other 3 base pairs here, and we have a tRNA, which brings amino acid number 5. And it works the same way. It's like a machine. Every time we have a new amino acid that enters that is corresponding to the 3 bases, the codon. And then we have a growing protein chain. This is a polypeptide, a long chain of amino acid. Questions about this part? OK. So about microRNAs, you heard a little bit about them, but just to freshen up your memory, these are DNA genes. And we have a typical gene. A typical gene forms a messenger RNA, mRNA, which then is transcribed to form a protein. OK? So far, you're following, right? But in some cases, we have RNAs, these are microRNAs, that bind to the messenger RNA to prevent formation of a protein. So the microRNAs, what do?
They are the ones that control gene expression.
They are the ones that, not only, but yes, they are ones that control gene expression. They repress gene expression. Okay? They're very, very important because since they're very small, they're short, it takes much less time to transcribe them than all a messenger RNA. So they work really quickly, and they are able to react in different stressful situations for the cell. For example, heat shock. If you take cells and you expose them to heat, they have to react really quickly, unless if they don't do it, they die. Okay? So microRNAs, some of them function in this kind of situations, but not only. Another Nobel Prize. So Andrew Fire was awarded the Nobel Prize for discovery of the RNA interference. So this is the mechanism of microRNAs, but other RNAs as well. And here you see how it works. So we have, this is the mRNA molecule. It is then exported to the cytoplasm, where it forms the protein. This is a growing protein. And what's the yellow part? What is the yellow? Ribosome, right. Okay? Adayonat. And here, in the other part of the cell, we have the microRNA, or the interfering RNA, which are also forming some complex in a protein called dicer, but this is not important for this class. And what they do is to repress the mRNA molecule. So they bind, this is an mRNA, the blue part. They bind to the mRNA and they chop it. Okay, and this way it won't form a protein. A little bit more about microRNAs. MicroRNAs, mirrors. We have a lot of microRNAs, more than 1,000. Actually, I think around 1,500 now were known. And they're very conserved. So there are microRNAs in humans, in primates, in rodents. This is a mouse. But also in other species like nematodes, okay, worms, for example. They also have microRNAs, less than we do, but they do have them as well. It is very, very conserved along the evolution. Now back to proteins. So this is a chain of amino acids, which is a protein. So this is a very short one, but this is just to give you an example. We have different 20 amino acids, and they are joined together to form this chain. Now, okay. I already said that we have 20 kinds of amino acids. Okay, and we have many, many different proteins. So the human has about 30,000 different proteins, but we have less genes than that. Okay, we have about 10,000 genes, but 30,000 proteins. So what does it mean if we have more proteins than genes? Think about it. Genes are, where are they? In the nucleus, in the DNA. Okay? And we have a lot of genes, but not as many as proteins. So this means that each gene can give rise to different proteins.
Effectivity.
Or?
Effectivity.
It's not effectivity, actually. It's more about modification of the protein. So for example, I think I have an example later on. I hope so. But this is called alternative splicing. It's like a Lego. Okay, so we have a chain of a protein. So imagine we have a Lego. So we have a red part, a blue part, a green part. And they can be, we can have a protein that contains all the parts. And we can have a protein that has only some of them. And this can be formed from the same gene. Okay, and this is how we have a lot of different proteins, but not as many genes.
What's it called? Alternative?
Splicing. Okay. The chain between the amino acid, the bond between them is called a polypeptide bond or a peptide linkage. Yeah. So the protein has to be folded. It has to be organized, not just to be a chain in the cell. It has to form some structure. And how does it happen? So here we have the amino acids. So they look just the same, but they're not. We have different amino acids right here with different characteristics. And what happens is that they're forming another organization layer. This is called secondary structure. Okay, so this part can be folded in like a zigzag, which is called beta-pleated sheet, or like a helix right here. This is alpha helix. So there are many different ways that this can happen, but these are the main ones, pleated sheet or alpha helix. And then each of these structures can be formed to a bigger protein. So here we have the alpha helix part, and here we have a beta pleated sheet part. And the protein usually contains more than one. Okay, so we have here beta sheet and another one and another one and another one. And here we have some alpha helix. And here we have an unstructured area, which is linear. It's straight. And usually proteins look like this. So this is called primary structure. This is secondary structure. And this is tertiary structure. And when we have some units like this, a few units like this, so in this case 2, so this is the final organization. This is layer 4. And this is the mature protein. Okay, so it's very complex, as you understand, but very, very organized. Okay? So we get from this sequence that is, remember how it forms? Each amino acid comes from what? From, right? That what happens there? From, so each amino acid is formed in the ribosome. Okay, it enters the chain in the ribosome with the help of the tRNA, and we have this chain, and then this chain is folding. So it starts in this layer, and it gets more complex and more complex. And finally, this is the mature protein. This is a protein in our bloodstream, actually.
So the amino acid is what's responsible for making that structure? Or
Well, it must be there in order to get a structure, yes. Yeah.
I'm sorry, just to get a broader picture. The point of this is the DNA is essentially sending out, I guess, kind of like messages for and these proteins then do things
Right,
according to the DNA's instructions.
right. So imagine what happens if we have a mutation. in our DNA. So this would not happen properly. We'll get there, I think, today. I'm not sure. OK, so this is an example of a protein, silk. OK, silk is a protein that has a lot of beta sheet in it. And this is why it's very, very strong. It's one of the strongest proteins in nature. OK, so mutations. Many, many neurodegenerative diseases, we're back to neurodegeneration, are due to different mutations. So mutations are genetic differences in our DNA that are not the inherited differences that give us different characteristics, OK? So it's in different areas of the DNA. And it's a change in the sequence of the DNA. OK, so in this case, this was, OK, so this is the original DNA sequence. So this is a double-stranded DNA. This is the 5 prime. This is the 3 prime. And this is the other 5 prime and the 3 prime. So it's anti-parallel. OK, it goes in this way. And this is a normal sequence. And we have different mutations. So this is an example of the protein that is transcribed from this sequences. Okay, so for example, AUG gives rise to a methionine. This is the first amino acid, the amino acid that starts each protein. Okay, and then we have threonine, histidine, doesn't matter the names. Okay, but each triplet, each 3 bases are a codon for a different amino acid. So this is the chain of the protein. So far, it's clear? Good. Now, this is an example for a mutation. So sometimes we have a substitution of one base. Instead of a T, we have an A here. OK? And then in the other strand, we'll get a U. And then we get a different codon. So this used to be ACC. And now it's UCC. And if you go back to the matrix, you'll see that it's a different amino acid. So instead of the pink one, which is called threonine, we get a different one, a yellow one, which is called serine. And if it's a different amino acid, what would happen then? Try to imagine. What would happen if we have a different amino acid in the protein?
That's a different protein.
Different protein is manufactured.
Right. So it could be a different protein. It can be a little bit different, and it can be a lot different. It depends where it happens. So proteins, you know, it's already very, very complex. Just try to turn off your phones, please. So it depends where this amino acid is. So in each protein, there are more important areas and less important areas. Why? So proteins can be responsible for cutting things, for example. And cutting things can happen in an active site of the protein. Okay, so each protein has some active site, almost each one. And the active site has the most important amino acids. So if there's a mutation that affects this site, maybe this protein will not be able to do whatever it has to do. But on the other hand, it can happen in a different area of the protein that would not affect the protein at all. Okay? So this is possible too.
Question.
Yes?
Mutations happen in our body all the time?
Yes.
Not just in nucleotides?
No. I'll get there. OK. Another kind of mutation is called frame shift. What is frame shift? So what happened here? Can you see? This is the sequence, and this is the new one. What happened here?
Skip the letter.
Look at the letters.
Skip, yeah.
Look more closely. So we have here an A, and suddenly this A is missing. And then what happened? Look at this sentence. So this sentence says, the fat cat sat. And suddenly we take out the T. And then we make another 3 pairs. And what happened to the sentence? It doesn't make sense anymore. And this is exactly what happened to the protein. Okay? So in this case, this is the start site AUG, which is correct. It's the same amino acid as we had here. But everything that is followed is completely wrong. And in this case, we absolutely will not be able to have a functional protein, okay? Because it's a totally different thing. It's a totally different protein, okay? So a frame shift mutation can happen due to two things: insertion of a base or deletion of a base. Okay, so if a base is deleted, for example, here it's 2 bases. So we had CT, GG, AG, and here we have CT and then AG. So 2 bases are missing, and that means that everything will be disrupted, okay? Because the codons, 3 bases that are forming the code, will just make a totally different code, okay? What happens here? Okay, so it's the opposite. We have insertion of 3 bases, TGG, and they also cause a frame shift. And then we have another different protein. So this is really, really important. So many neurodegenerative diseases are caused by genetic mutation. For example, Huntington's disease. So this is an inherited disease, a genetic disease that is caused by an expansion, a growing area that has a lot of CAGs in it. So we have a lot of repeated CAG sequences in a certain area of the genome. So we have CAG, 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 CAG. And all of us has the CAGs, but some people has more than others. Okay? So in the normal case, you will have up to 28 repeats. So you have, nothing happens. This is normal. If you have up to 28 repeats, so some have 27, some have 28, some have 25, but this is all normal. But when you have more than that, so when you have between 28 and 35 repeats of CAGs, this is classified as an intermediate case, but still it's usually not that terrible. So up to 35, you can live with that normally. But up to 35, no. No. But more than 35 cause a disease. So up to 40 is like a moderate case of the disease, and more than 40 is a very serious disease. So it's basically a toxic protein. So we have a lot of CAG. CAG is the code for glutamine. So this is amino acid that is called glutamine, and we have a lot of glutamines in the protein. And when we have too many of them, it is just toxic to the cell, and cells start to die. And which cells are more susceptible and are more sensitive? Neurons. Cells in our brain are more sensitive. And this is why we get a neurodegenerative disease, and you'll have a whole class, I think, about Huntington's. This protein, by the way, is called huntingtin. Okay? This is another example. So Alzheimer's disease. We have different mutations that are causing what is called neurotoxicity. So here, for example, this is the protein that cuts the APP. APP is amyloid precursor protein. And then it makes the A-beta, amyloid beta. Okay? And when we have a mutation here, this is the Swedish mutation. I think it's one of the most studied mutations in neurobiology. So when we have this mutation, the processing of the amyloid protein is a little bit different, and it causes neurotoxicity. This is a different mutation. It's called the PS1 presenilin 1, and it's a different mutation. Later on in the pathway, it is right here. And it gives rise to a different process that does not occur properly. But all of these mutations give rise to Alzheimer's disease. So you see, this is a very, very sensitive system. Each mutation in the pathway will still give the same phenotype, okay, the disease, the Alzheimer's disease. Okay, so other neurodegenerative diseases, like prion disease. Have you heard of the mad cow disease? Yeah. Okay, so the mad cow disease, it's called Creutzfeldt-Jakob disease. And there are other diseases like fatal familial insomnia. They occur because proteins are not folded correctly. Okay, so you already know that a protein is a long chain and it has to be folded in a very specific way. But if it does not occur, like here, okay, so we get this long protein, it's like a polypeptide that is not folding. And then it becomes a plaque. Okay, this is called a plaque, like amyloid plaques. So we have plaques of different proteins. And this can be very, very toxic. Okay, a little bit about heredity. So what is that? This is the passing of genetic factors, genetic characteristics from a parent to the child. So in this case, this is the mother that carries some mutation. And in this case, you see they have 4 children. But what happened to the gene? This is the red one. It passed only to girls in this case. Okay? So this is a male and this is a female. And then this girl had 2 children and passed the gene, the affected gene, to her offspring. So this is a little bit about heredity. And we have different types of heredity. So in this case, this is recessive heredity. So this means that it will not pass, the feature will not pass necessarily, okay? So that means, this is like blue eyes, for example. So you know that if your parent has blue eyes, maybe you will have, but more likely you won't have blue eyes unless the other parent also has blue eyes. Why is that? This is because it's a recessive gene. So this means that the chances of getting blue eyes, for example, are a little bit lower. So how does it work? You wanted to ask something?
Yeah, I want to know where the man before got the recessive gene from.
Where?
Was it a mutation, or where did they get it from?
So in this case, this is just an example. It's not a mutation. It's a gene.
Okay.
A mutation is a different thing than a gene. Okay, so mutation is something that, okay, so we have SNPs, single nucleotide polymorphisms, that are like signs in our DNA that are different between me and you, for example. And there are SNPs that are normal and they're not a mutation. A mutation is something, and by the way, the SNPs are more than 1% prevalent. When it's less than 1%, it is defined as a mutation. And usually mutation can be silenced, okay, and then nothing happens. And if it's not silenced, then sometimes we get some phenotype. So I will give you some examples which will make it a little bit clear. This is a dominant heredity. So in this case, this parent, for example, has dark hair, okay, and this doesn't. And it passes on the dark hair to the offspring. So in this case, all the 3 out of the 4 children, okay, this is the one that doesn't have the dark one, okay? They got, what, 6? Oh, yeah, right. Okay, but it doesn't matter. So they got the dominant gene. By the way, Huntington's disease is dominant. Okay, so this is an example for a dominant gene. So now everyone usually tries to do that in the class. So you're trying to roll your tongue. Some can, some cannot. This is a dominant feature. So if you can, most likely your child will be able to do that too. Okay. This is another example. So this is a recessive gene. Can you do this thing? I cannot do that. Okay? Right. So you can.
It's genetic?
Yeah. Everything is genetic. Okay. Curly hair. Obviously, it's a dominant gene. And also freckles are. Freckles, nemashim. Okay, so this is also a dominant gene. This is the Birkat Kohanim that you're probably familiar with. So most of the people can do this, but some cannot. Okay?
I thought it was meant more like a muscle that you have to train.
No.
It's not like eyebrow disease.
No. No, no, no. Okay. Free earlobes. Okay, another dominant feature. So some people have connected earlobes. Okay? And this is recessive. And cheek dimples. Okay? They're also dominant. So these are examples that make it a little bit also dominant. Also dominant. And this is called widow peak. You know this part? Like Mickey Mouse? This is another dominant feature. And blue eyes, the classic example. So this is a recessive gene. So a little bit more about genetics. In biology, when we have a gene that we want to study, so Mona already told you that, but maybe you forgot, what we do is to engineer a mouse that expresses this gene in high levels. We call it transgenic mouse, okay? And it helps us to develop models for diseases or to study functions of genes. And what we do is to change the genome of the mouse. So sometimes we take a human gene and we put it inside the mouse genome. And then we are able to study a human gene inside a mouse model. Okay, so we can make transgenic mice or transgenic other different animals, but usually it's mice. And then they can develop different characteristics. For example, we have a mouse that is transgenic for the human APP gene, amyloid precursor protein gene. And what happens to this mouse is that at a certain age, it starts to develop Alzheimer's disease-like features. So what happens is that it aggregates plaques, amyloid plaques. It accumulates these plaques in the brain, and it starts to forget things. So when we put it in a maze, the mouse is unable to find its way. Okay, so this is a very classical example for a transgenic mouse model. This is a very old model. It is still studied, and it has advantages and disadvantages like everything. But this is a very important tool in biology. So this is an example of another transgenic mouse. So this mouse expresses a protein that is fluorescent. It's called GFP. It's a green fluorescent protein. And usually we just don't make glowing in the dark mice. But what we do is to make a transgenic animal and tag this protein that we're interested, the gene we're interested in, to a fluorescent protein. So for example, we want to see where amyloid protein, amyloid precursor protein is. So we tag it to a fluorescent protein, and then we're able to see it. Okay, we can see where it is in the brain, for example. Okay, so this is another thing that allows us to study location of proteins and what exactly happens there. It's a very, very important thing. Generally speaking, disease models are very, very useful for medicine, obviously. So it's for a disease model, these are the examples I gave you, but also for transplantations, okay, for nutritional supplements, for pharmaceuticals, and for human gene therapy. It's also very useful for agriculture. So it's more common in transgenics, not of mice, but of plants. For example, if we have a corn plant and we want it to be resistant to some disease, so we can manipulate it and we can make a plant that is resistant to the disease. And this is also used to make a plant that is growing better or that makes fruit that looks better or smells better or lasts longer, for example. Okay? And it is also used in the industry. So a very classical example is the spider goats. So these are transgenic goats that were engineered that they will produce a silk in their milk. Yeah. Why? So why do we need silk? It's expensive. Yeah, people want it for clothes. So what? right? So what someone did is to clone it in the goat, and then the goat produces milk with the silk. And then the silk can be used for other things.
What happens if baby goats, are they able to process the silk?
What do you mean? When they're drinking milk?
Yeah.
I guess they used foster mothers. Yeah, it's an interesting question. Okay, so just some questions to end this class. So which structure is represented here in the figure? It's a protein. This is a chain of a protein. And which structure of the protein is that? The basic, the complex. Remember the degrees of complexity?
Basic.
Right. So this is the primary structure, the basic one, the first layer. Okay? And here we see amino acids. Okay. What is this? What do you see here? That's a little bit folded protein. So here you see, what is this? Helix, yeah. This is an alpha helix, and here is another one, and here is a loop here. And what are these? Yeah, so this is not linear. This is actually how we represent beta sheets. So these are the zigzags. Remember the zigzags? So this is how we draw them usually when we are referring to them in a bigger molecule. And these are unstructured areas here, and here, and here, and here. What is this?
Protein.
Yeah, this is a protein. So this is also a specific protein. This is hemoglobin. This is the protein that carries oxygen in our bloodstream. And these are basically 4 units of a protein. So each of these has alpha helixes and beta sheets in it. And 4 of these subunits form the whole protein, the entire protein. Which one is the correct one? Look closely. Okay. So look at the bases, look at the direction. Let's try to eliminate some.
It can't be D.
It can't be B. D. Okay, let's start with D. So we have here 5 prime and here another 5 prime. Can they form a structure?
No.
A duplex? No. Okay, this is upside down, one of them, in the opposite direction. So it's not D.
And B also because A is going with A.
Right. A and A cannot bind to one another, right?
Yeah.
Okay. So it's not B. It's not D. So which is the correct one? So actually, it's C, but I don't remember where is the base that is not correct. But somewhere here, there is a base that cannot pair to the other one. You can find it, but I'll just tell you that this is the situation here.
Something about the flip.
They're both of them.
No. One of them is not correct.
Position of the 5 or the 3.
No, the 5 and the 3 are okay.
Yeah, but in A and C, it's different.
It's exactly the same.
It's the same sequence. Okay, so it's not this example then. Okay. Oh, okay. So look here. What is the difference between A and C?
The direction of the first strand.
Right. So this goes from 5 prime to 3 prime, and this one starts in 3 prime. And what did we say? That we start with 5 prime. So it's A. We have another example. I'm not sure if I put it. Yeah, these are the. Okay, right. So it's not here, but if anyone is interested, I'll put it in the website. Now here you have 2 different DNA sequences. And you see this is a graph with increasing temperature. Okay, so we're increasing the temperature. We put the DNA sequences in there. What can you say about the sequences? Okay, this is the denaturing fraction. So it means which fraction, which amount of the protein is not folding properly, is denaturing. What can you say about these 2 sequences?
One has a lower temperature.
One has a lower temperature, right, exactly. So which one? Obviously this one has a lower denaturing temperature, and this one has a higher. What would you conclude about the sequence?
The bonds.
The bonds, right. So Which bases form stronger bonds? C and G, right. So we have more GC content in this sequence. This is why it denatures in higher temperature. Is this clear? Good. Okay, so I think we're done. If anyone has any questions, just ask. And have a good weekend.